welcome to this video for Advanced Higher Biology. This is Unit 1, Cells and Proteins. We're looking at Key Area 1, Laboratory Techniques, and specifically today focusing on protein detection. When we consider protein detection, we are mostly using immunological techniques, and these are used to detect and identify proteins. What's expected from you is to be able to describe what we mean by an immunoassay technique, to be able to name the type of antibodies used in this technique, describe the chemical labels that are used in this technique, and explain when the Western blotting technique would be used and describe the process. Now, when it comes to detecting specific proteins, previously we have looked at separation techniques, so how we would separate proteins from each other or from a mixture that they happen to be in. When it comes to detecting which proteins we have managed to isolate, one of the main techniques we use is antibody and antigen binding. And this is what we mean by an immunological technique. Obviously, you should be aware, if you've done higher human previously or from recent events, that antibodies and antigens are highly used within our immune system. And they are both types of protein, and we can sort of harness their capabilities in order to detect our specific proteins. Overall, essentially, an immunoassay technique is using the ability of antibodies to bind to specific antigens. An antibody is a Y-shaped globular protein, and that is produced as part of our immune response. When we look at antibodies, they have what we refer to as very specific antigen binding sites. If we look at their shape, they're overall Y-shaped globular proteins with these particular areas here, which means they will bind to a specific antigen. The other antigens will not be bound by that antibody. So those ones there would not be bound. Only the ones with the correct shape can bind. It's very similar to an enzyme and substrate binding process. Now, when we form antibodies naturally, they will take several different shapes to bind to different parts of the antigen. So we would normally have several types of antibody with various shapes which are complementary or specific to the antigen, and that means these ones are polyclonal. So these ones here are our polyclonal antibodies. On the other hand, we have monoclonal antibodies, and these tend to be the ones which are produced in the lab. Now, these have the same level of specificity, so we know they are precisely specific to one particular antigen. And these are referred to as our monoclonal antibodies, as they will only bind one specific part of the antigen, whereas our polyclonal antibodies produced naturally will bind to more than one part of the antigen. When we use these in the lab in order to detect our specific antigens, what we do is we use a monoclonal antibody that has the enzyme attached. That particular enzyme is capable of having a colour change reaction, and that's used to detect and also potentially quantify the presence of a specific antigen. So if this is our antigen here, and our antibody, this is a monoclonal antibody, so it's specific to this particular area of our antigen. It has its reporter enzyme bound onto it, and as a result, when that antibody binds to the antigen, the reporter enzyme reacts. It's usually a colour change, could sometimes be fluorescent. What we can then do is use that reporter enzyme to detect the presence of the antigen, but also we could perhaps be using a colorimeter or some other kind of measuring technique in order to identify just how much, so quantifying the presence of our particular antigen. If the antigen itself isn't present, then the antibody can't bind and the enzyme is washed away before any reaction will actually take place. And it's important to just be aware that in addition to enzymes, these reporters could take the form of chemicals which luminesce or fluorescent chemicals. Now, in some cases, we will use the assay itself to have a specific antigen, and in this case we're trying to detect the presence of antibodies. In this case, you would have your antigen, your specific antigen, would be bound to a solid material, so it's a gel or an amino acid plate, and the sample itself would be washed across, and we would be looking for the presence of specific antibodies. This is obviously a very basic example of this setup, and what you would notice is that we have our antibodies have our reporter enzyme attached to them. Now, if this was a case where we were trying to, for example, to detect a disease presence, we're looking to detect a specific antigen, what we're looking to do is identify whether or not our individual patients have antibodies present in their blood, which would indicate that they've already been exposed to the particular antigen. If that's the case, obviously, we don't necessarily have reporter enzymes bound to the natural antibodies that would be produced by the patient. So in this case, we use a slightly more complicated technique where we use a secondary antibody involved. And this is the similar process to what is used in the detection of HIV. So the immune system of a patient with HIV will have produced antibodies which are complementary to the HIV viral proteins. And if we can detect those antibodies, that would indicate a positive result. In this case, our HIV protein, so the viral protein, is here. It's attached to a solid surface, and we're then adding the patient sample. So in this case, we've added the patient sample, and the antibody in the patient sample has bound to our viral protein. The issue we have with that is there's not necessarily a detectable label attached to that naturally produced antibody, which was produced by the patient. Consequently, we then need to use a secondary antibody, which does have a detectable label attached. And that's going to be complementary to our patient antibody. And that's added and will indicate if our patient has HIV. Obviously, if the patient does not, then they will not have any of those antibodies present. So they won't bind to the viral protein, and the secondary antibody won't have anything that it can bind to. Consequently, the detectable label won't react, and we wouldn't be able to identify it. We can also use Western blotting in order to detect or identify specific proteins. And Western blotting is a protein detection technique which is used after separation by the STS page technique, which we spoke about in the last lesson. So after our proteins have been separated by gel electrophoresis, so they've gone through our separation process, we've had our negative electrodes and our positive electrodes attached to the gel, the proteins have washed through the buffer and they've slowly separated out. Now that means they've become separated from each other, but what we might need to do is detect them and figure out exactly which proteins it is that we have within that sample. What we then do is we take the gel and it is transferred or blotted onto a solid medium, and that's more convenient for our further study. So the gel itself is really soft and flexible, quite easy to break, and will dry out. So what we do is we transfer that, blot it onto a more solid medium, some kind of membrane usually, and that just makes it more robust and easier to study in the future. Once we've done that, if this is our more solid medium down here with our proteins attached, we can then, in a very similar process, we're going to flood it with our fluorescent labelled monoclonal antibodies or other reporters. If those antibodies are then able to bind to the target proteins, and then any excess is washed off. Obviously, again, if they are unable to bind, if the protein isn't there, they will not bind at all, so there will be no signal. You can then visualise this underneath a specific wavelength of light, and any fluorescent labelled antibodies will allow the location and the identification of specific target proteins. So from that, you should understand what we mean by an immunoassay technique, name the type of antibodies we've used in this technique, describe the chemical labels we use, and explain when the Western blotting technique would be used and describe the overall process. As ever, please feel free to get in touch if you have any questions